application of the DILIsym model, where we use in vitro data inputs to make in vivo extrapolations in predicting drug-induced liver injury, or DILI. The DILIsym model has been designed to assist in evaluating hepatotoxicity risk for compounds throughout the clinical development pipeline, from in vitro type experiments on the preclinical end, all the way through clinical trials, in vivo animal models, even post-marketing. We have a number of applications of DILIsym in each of these areas of the clinical trial pipeline, the clinical development pipeline. And for this particular presentation, we'll be focusing on the in vitro preclinical area. Before we dive into the application, I'd like to use this opportunity to give everyone a very brief overview of DILIsym. It is a mathematical model of drug-induced liver injury, and we make these predictions in four species: the human, the rat, the mouse, and the dog. We have kind of a baseline representation of each, representing, say, the average human, the average rat. And we also have our SimPops, or simulated populations, that allow us to account for inter-individual variability. Within the model, we have captured the various cellular processes that are essential to representing drug-induced liver injury. This summary diagram shows the major submodels and how they interact. And if you start in the upper left-hand portion, you can see that we make predictions of exposure of the parent and various metabolites, and how they interact with the key hepatotoxic processes like reactive oxygen species production, mitochondrial dysfunction, bile acid transport, and then how those all interact with the hepatocyte life cycle to initiate apoptosis and/or necrosis. The innate immune system can participate in exacerbating or ameliorating injury. As well, the innate immune system can help drive a proliferative response to offset the hepatocyte loss due to injury. And then there's a variety of biomarkers of injury that are also represented in DILIsym. These qualitative connections are made quantitative by using exemplar compounds, and we have a number of examples of these exemplar compounds in kind of each of the key mechanistic areas: reactive metabolite, mitochondrial dysfunction, bile acid transport or inhibition, as well as exemplars that incorporate multiple mechanisms in their hepatotoxicity components. DILIsym is really best described as compartment-based modeling. It's a deterministic model utilizing ordinary differential equations. As well, it includes a form-to-function connection, wherein the loss of hepatocytes due to predicted injury feeds back and maintains mass balance such that the remaining, for example, drug can only be distributed or taken up by the viable remaining hepatocytes. That form-to-function connection is unique for a toxicity type model such as DILIsym. So now that we have a brief overview of DILIsym, I'd like to now go into the application example. And this is focused on a drug known as methapyrilene. It's an antihistamine and a sedative that was available on the market into the 1970s or so, when it was withdrawn due to some examples of carcinogenesis and drug-induced liver injury in preclinical rat models. As you know, these injuries were detected, these liabilities were detected, there was a little more increased study, and it was determined that it was really the injury, the drug-induced liver injury, was driven by reactive metabolites, and it was largely periportal in origin. Interestingly, there was no drug-induced liver injury whatsoever reported in humans. And so we seem to have a bit of a species difference. So we undertook this simulation application project really using exclusively in vitro data, and then making predictions of in vivo hepatotoxicity. So for the physiologically based pharmacokinetic modeling to predict hepatic exposure, we used log P and pKa, so physical chemical properties of the compounds. We had access to some metabolic partitioning data in rat and mouse hepatocytes, covalent binding data in rat, mouse, and human microsomes to give an indication of the rate of reactive metabolite generation. And then we evaluated the impact of all this data and the ranges throughout on the subsequent simulations, such that this ends up being a nice application of the IV-IVE sort of scenario, as well as providing some validation of DILIsym in that these simulations were not used to optimize the underlying model parameters in any way. So pulling these parameter values together, our initial simulation in the baseline simulated rat was that there in fact were predicted increases of ALT, pretty substantial increases up to 500 times the baseline ALT values at a dosing range with a threshold across the dosing range of 150 to 200 milligrams per kilogram QD dosing over 3 days. Within the literature, there is actually combining the various reports. The simulation results are actually quite consistent with the observed threshold response, in that it also appears that in the rats in the preclinical studies, that same threshold of 150 to 200 milligrams per kilogram seem to exist. So our simulation results really seemed quite consistent for rats. Carrying forward the species difference theme, interestingly, we did not predict, or DILIsym did not predict, that there were any increases in ALT or injury in mice, and those species differences were based on those in vitro data of the types that I described just a moment ago. And so really, using these rat data and the mice data, we are effectively predicting the in vivo response in these preclinical animal models based exclusively on the in vitro data. Carrying this forward to the clinical predictions of clinical hepatotoxicity, in fact, as the graph shows clearly, there were no increases in ALT over baseline across a wide range of methapyrilene doses. And again, this is consistent with the fact that there were no reports of human hepatotoxicity due to methapyrilene use. And it also, kind of from the application point of view, again, proves that with DILIsym, you're able to use in vitro data to make predictions of clinical trial results. Of course, additional data increases confidence in predictions, but this is an excellent starting place for a rather limited data set. So this is kind of the set of simulations with our baseline average human, baseline average rat, baseline average mouse. But to really fully assess hepatotoxicity risk, we need to consider that interpatient, inter-animal variability aspect. So we then conducted these similar set of simulations in our SimPops, accounting for variability in a variety of relevant biochemical areas, and in rats, mice, and humans. And the figure here shows that in fact, we did predict a range of responses as far as the maximum ALT is for rats given a single 600 milligram per kilogram dose. However, there was no predicted increase whatsoever in ALT at this dose in humans nor mice. So again, it's consistent with what was observed, that there were species differences. Even considering inter-individual variability in some of these key areas of the underlying hepatic biochemistry, still no predictions of clinical hepatotoxicity, yet consistent or appropriate predictions of preclinical rat hepatotoxicity. This work was written up and published in a paper published in 2012, and it really, again, establishes the fact that DILIsym can be utilized for in vitro to in vivo extrapolation applications in this case, including reactive metabolites. Thank you for your attention.